Welcome to our deep dive into the fascinating world of biotechnology. It's a field that's all about using living things and their amazing abilities to develop new technologies and products that can change our world for the better. We're talking about the ability to manipulate DNA, the very blueprint of life. Get ready for a wild ride as we uncover the science behind cloning, gene editing, and so much more. Imagine you have a recipe for the most delicious chocolate chip cookies ever. You want to share it with everyone, so what do? You make copies. That's kind of what DNA cloning is like, but instead of cookies, we're talking about genes. Scientists use special enzymes like tiny molecular scissors to cut out a gene of interest from one organism, then they paste that gene into a different organism, often a bacterium. Why bacteria? Well, bacteria are like the little factories of the biological world. They can read DNA and use it to make proteins, which are the building blocks of life. So by inserting a gene into a bacterium, scientists can trick it into making lots and lots of copies of that gene. But it's not just about making copies. Remember, genes are like recipes, and they contain the instructions for making proteins. So by cloning genes, scientists can produce large amounts of specific proteins that have important applications in medicine, agriculture, and other fields. Think about insulin, a life-saving hormone for people with diabetes. In the past, insulin was extracted from animals, which was expensive and not always reliable. But now, thanks to DNA cloning, we can insert the human insulin gene into bacteria and let them do the work. These bacteria become tiny insulin factories, churning out large quantities of this essential hormone that is identical to what humans produce. This breakthrough has revolutionized diabetes treatment and improved the lives of millions. And that's just one example. DNA cloning is also used to produce other important proteins like growth hormones, antibodies, and enzymes used in industrial processes. It's like having a biological toolbox that allows us to harness the power of nature for the benefit of humankind. DNA cloning also plays a crucial role in gene therapy, a cutting-edge field that holds the promise of curing genetic diseases. In gene therapy, scientists use modified viruses as delivery vehicles to introduce healthy copies of genes into cells with faulty ones. These healthy genes can then replace or compensate for the malfunctioning genes, potentially correcting the genetic defect and treating the disease at its root. So, DNA cloning is not just about making copies, it's about harnessing the power of genes to produce life-saving medicines, develop new therapies, and advance our understanding of the living world. Now, let's talk about a technique that's so important it's hard to imagine modern biology without it. Polymerase chain reaction, or PCR for short. If DNA cloning is like making copies of a whole book, PCR is like making copies of a single page. Imagine you have a tiny drop of blood at a crime scene. How can scientists possibly get enough DNA from that tiny sample to identify the suspect? That's where PCR comes in. PCR is like a molecular photocopier for DNA. It allows scientists to amplify or make millions of copies of a specific DNA segment from a tiny starting sample. This is incredibly useful for a wide range of applications, from diagnosing diseases to solving crimes. So how does PCR work? Well, it relies on a special ingredient called DNA polymerase, an enzyme that naturally copies DNA in our cells. PCR basically hijacks this enzyme and puts it to work in a test tube. The process starts by heating up the DNA sample, which causes the double helix to unwind and separate into two single strands. Then, short DNA sequences called primers are added to the mix. These primers act like little bookmarks, attaching to specific regions of the DNA that scientists want to copy. Once the primers are in place, DNA polymerase gets to work, using the primers as starting points to build new DNA strands that are complementary to the original template. This cycle of heating, cooling, and DNA synthesis is repeated over and over again, with each cycle doubling the amount of DNA. After just a few hours, a single DNA molecule can be amplified into millions or even billions of copies. Now, with enough DNA on hand, scientists can analyze it using various techniques like gel electrophoresis, which separates DNA fragments by size, creating a unique DNA fingerprint. This ability to amplify DNA has revolutionized fields like forensic science. Remember that tiny drop of blood from the crime scene? With PCR, scientists can now analyze even the smallest traces of DNA evidence to identify criminals and solve crimes that were once unsolvable. PCR is also a game changer in disease diagnosis. By amplifying DNA from a patient's blood or tissue sample, doctors can detect the presence of viruses, bacteria, or even genetic mutations that cause diseases like cancer. This is particularly important for diseases like HIV, where early detection is crucial for effective management. We've talked about copying DNA and amplifying specific segments, but what about reading the actual sequence of letters that make up our genetic code? That's where gene sequencing comes in. Imagine you have a giant library filled with books written in a language you don't understand. Gene sequencing is like having a magical translator that can decipher the language of DNA and reveal the secrets hidden within our genes. Gene sequencing is the process of determining the precise order of nucleotides, adenine, guanine, cytosine, and thymine, in a DNA molecule. It's like taking a single strand of DNA and reading it letter by letter, revealing the genetic blueprint that makes each of us unique. Early gene sequencing methods were slow and laborious, but thanks to incredible advancements in technology, we now have next-generation sequencing technologies that can sequence entire genomes in a matter of hours or even minutes. The ability to sequence DNA quickly and affordably has revolutionized fields like personalized medicine. By analyzing an individual's genome, doctors can identify genetic variations that may influence their risk of developing certain diseases or their response to specific medications. This information allows doctors to tailor treatment plans and preventive strategies to each patient's unique genetic makeup, leading to more effective healthcare and better outcomes. Gene sequencing is also a powerful tool for understanding and treating genetic diseases. By comparing the genomes of individuals with and without a particular disease, scientists can identify genetic mutations that may be responsible for the disease. This knowledge can then be used to develop gene therapies that target the specific genetic defect, offering hope for cures to diseases that were once considered untreatable. But gene sequencing's applications extend far beyond medicine. It's also used in evolutionary biology to trace the ancestry of species and understand how life on Earth has evolved over millions of years. So, gene sequencing is like having a powerful lens that allows us to peer into the intricate world of DNA and unlock the secrets of life. Now get ready for this, science fans, because we're about to enter the world of gene editing. And no, we're not talking about science fiction here. We're talking about CRISPR, a revolutionary technology that's changing the way we think about treating diseases and even engineering life itself. Imagine having a tiny pair of molecular scissors that can precisely cut and paste DNA, allowing you to correct genetic defects, insert beneficial genes, or even disable harmful ones. That's essentially what CRISPR allows scientists to do. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, which is a mouthful, I know. But basically, it's a system that bacteria have been using for millions of years to defend themselves against viruses. Think of it like the bacteria's immune system. When a virus invades a bacterium, the bacterium can capture snippets of the virus's DNA and integrate them into its own genome, creating a sort of genetic memory bank. These stored viral DNA fragments then serve as guides, allowing the bacterium to recognize and target the virus if it ever tries to invade again. It's like the bacterium keeps a wanted poster of the virus's DNA, so it can quickly identify and eliminate the threat. Scientists have harnessed this bacterial defense system to create a powerful gene editing tool. By designing guide RNA sequences that match a specific DNA target, scientists can direct the CRISPR-associated protein, Cas9, to cut DNA at that precise location. Once the DNA is cut, the cell's natural repair mechanisms kick in, attempting to fix the break. Scientists can exploit these repair pathways to introduce changes to the DNA sequence, such as disabling a harmful gene, correcting a mutation, or even inserting a new gene altogether. The precision and efficiency of CRISPR have made it a game-changer in various fields, particularly in medicine. Scientists are now exploring its potential to treat a wide range of diseases, from genetic disorders like cystic fibrosis and sickle cell anemia to infectious diseases like HIV and even cancer. Imagine a future where we can use CRISPR to correct the genetic mutations that cause cystic fibrosis, allowing patients to breathe freely for the first time. Or, imagine using CRISPR to edit the DNA of immune cells, making them more effective at recognizing and destroying cancer cells. These are just a few examples of the incredible potential of CRISPR in medicine. While there are still challenges to overcome, such as ensuring the safety and ethical use of this technology, the promise of CRISPR to revolutionize healthcare is undeniable. But CRISPR's applications extend far beyond human health. It's also being used in agriculture to develop crops that are more resistant to pests, diseases, and harsh environmental conditions. CRISPR is also a valuable tool for basic research, allowing scientists to study the function of genes and unravel the complexities of biological systems. From cloning and PCR to gene sequencing and CRISPR, these tools are revolutionizing fields like medicine, agriculture, and industry. The possibilities of biotechnology are limitless.